I'm Rick Godley, and I'm representing the Migraine Science Collaborative. And I'm grateful that Dr. Pietrobon has volunteered to be the first scientist to allow me to practice my interviewing skills. This all started because I read an article of yours recently in Science Advances called Astrocyte Dysfunction Increases Cortical Dendritic Excitability and Promotes Cranial Pain in Familial Migraine. So I now am asking Dr. Pietrobon to help me understand the significance of this research and how she accomplished it. Daniela Pietrobon performed her early scientific training at the University of Padova, Italy. She completed her postdoctoral fellowship in the Department of Membrane Research at the Weizmann Institute of Science in Israel. Her postdoctoral fellowship followed at the Harvard Medical School in the Department of Cellular and Molecular Physiology. She returned to the University of Padova, where she has held various positions of responsibility for research and education. She is currently professor of physiology within the Department of Biomedical Sciences at the University of Padova. Along the way, she has studied and published extensively on the topics of genetic mutations, causing a rare form of migraine with aura, and resulting in excessive glutamate release, and increased susceptibility to cortical-spreading depression. Mitochondrial function and ion channelopathy, particularly calcium channels and more, are also of interest to her. So with that introduction, I want to start with the conclusion of your paper and allow you to try to explain it in your own words. And here it is, the words that were printed. Our results demonstrate that impaired astrocyte function alters neuronal activity in the cingulate cortex and facilitates migraine-like cranial pain states in a mouse model of migraine. So if we were just sitting together having our cup of coffee and I have my, my cup here, I'm hoping that you could try to explain to me what that all means. You want to give it a try? Thank you.
Let's try. So first of all, let's talk about the mouse model we have investigated, which is a genetic mouse model of a rare form of migraine called the familial hemiplegic migraine type 2, which is a monogenic form of migraine. So it means that it is caused by mutations in a specific gene. And this gene, in this case, encodes a protein, the alpha-2 sodium potassium ATPase, that is expressed almost exclusively in astrocytes in the adult brain. And this protein is fundamental for important astrocyte functions, such as, which includes the rapid clearance of glutamate released during neuronal activity, rapid clearance of glutamate, which is fundamental for the correct brain function. And this is performed by astrocytes thanks to this protein, which is mutated. And this protein, which is mutated in FHM2, as I will call now familial hemiplegic type 2, is fundamental to allow this rapid clearance of glutamate, which occurs because there are specific glutamate transporters in the astrocytes, but the energy for this glutamate transport is provided by this pump. And it is also fundamental for another important function, which is keeping potassium homeostasis. So keeping the physiological concentration of potassium in the extracellular space. And also this is obviously very important for the correct brain function.
Okay,
now,
interrupt you for a second.
Yes.
So the, in a really simplistic way of thinking about this, one of the functions of the astrocytes is really monitoring and cleaning up these neurotransmitters, glutamate, which if it's not done, it's just they continue to stimulate the cells. So it's a way of resetting the stage for the next time that a release of glutamate is needed.
Exactly.
Is that right? And is this?
And it prevents the excessive increase of glutamate, which is dangerous. It leads to the neurodegeneration. But it's obviously, besides that, it is fundamental for the correct transfer of information, as you say, because it resets the synapses for the next action potential and the next release of glutamate.
And by the same token, controlling potassium is important because it's helping control the cell electrical signals that are carrying the messages along too.
To prevent the depolarization of neurons.
And this mutation is the one that it occurs in people too, as well as in this mouse model, right?
No, this mutation occurs in people, and we introduced the mutation found, one of the mutation found in people, in patients with this rare form of migraine, into the corresponding gene in the mouse, thus creating a mouse model of the disease. And what the mutation does to this protein, it produces loss of function. It inactivates the protein. So, and we have shown, we have the mouse model that we have is an heterozygous mouse model. It means that this model, this mouse has 50% as a reduction of these sodium potassium ATPases in the astrocytes. They are reduced to 50% than the normal, the wild type mouse. And we have shown that then, as a consequence of this, both the glutamate and potassium clearance are impaired in this mouse model. So we impaired in the sense that the rate of clearance of glutamate and of potassium from the extracellular space is reduced in the model as a consequence, because they have 50% of the ATPase. And so the mutation, and we have shown this in a previous study, that this occurs. We've shown this in a previous study of my lab, in the somatosensory cortex. And we've shown that as a consequence of this results in a facilitation of induction of cortical spreading depression. Now, in this study, we have shown that this occurs also in the cingulate cortex, and this results in facilitation of migraine-like cranial pain, as I will explain in a moment. So obviously the same defects in different cortices give rise to different defects and different aspects of the migraine pathogenesis. Why cingulate cortex? Because in migraineurs, it has been shown that infusion of nitroglycerin, which is an NO donor, induces a delayed migraine-like headache with associated features such as premonitory symptoms and also allodynia, so a condition in which pain is caused by a stimulus, which is like a light touch that does not normally elicit pain. So this nitroglycerin infusion induces in migraine a migraine-like attack, okay? And very interestingly, the cingulate cortex is among the regions that are activated both during spontaneous migraine attacks in migraine and in the premonitory phase of this delayed migraine-like headache that is induced by NO infusion. In animals, nitroglycerin, which generates NO infusion, lowers the mechanical nociceptive threshold. So in other words, it induces mechanical sensitization, and it induces a nociceptive response to a stimulus that should be innocuous, which is innocuous, does not elicit pain if NO is not infused. Quite interestingly, in the genetic mouse model that we are studying, infusion of NO of nitroglycerin at a concentration that does not do anything in wild type, does not produce any effect in wild type mice, it triggers orofacial pain in the genetic mouse model, which therefore show hypersensitivity to a migraine-like trigger. This has been shown by Jennifer Romanos and Mirko Santello, who are the first and last author of this Science Advance paper. And this is very interesting finding because it shows that these mice also model, in a sense, the nitroglycerin-induced allodynia observed in migraineurs. And the very interesting finding, then they went on to study the mechanism underlying this hypersensitivity, and they showed that this is due to the impairment of glutamate clearance by astrocytes, specifically in the cingulate cortex, and the consequent increase in firing activity of pyramidal neurons in the cingulate cortex. Then perhaps I will come back to this, but this is the essential message.
So I have a question about the allodynia. So allodynia, it can be both associated with a cortical spreading depression as sort of almost part of the aura, but it also is associated with central sensitization, which is a chronic form of migraine. And the fact that you were looking at the orofacial region is particularly interesting to me because I'm interested in this phenomenon that I see clinically where people develop a sense of pressure and pain and other neurologic conditions without even triggering, or maybe there's some triggering of it, but it almost seems spontaneous. So your findings begin to beg the question of, are we starting to understand why there is this sort of spontaneous symptom of migraine, which does not involve perhaps a cortical spreading depression?
No, because there are many aspects. I think cortical spreading depression is fundamental to explain migraine aura. Obviously, it underlies migraine aura, and it may be fundamental to explain the onset of a migraine attack if you believe that migraine aura is the phenomenon that, as has been shown in animal studies, activate the meningeal trigeminal nociceptive afferents that innervate the meninges, and which is, it is thought that activation of this is the beginning of migraine pain. But then the prolonged migraine pain is due to sensitization, both peripheral and central. And the question is, and what this paper also give a contribution, because the question is, central sensitization, which is a form of, probably is due to synaptic plasticity. Is a form of memory for, unfortunately, pain memory, but so it's a long-lasting change in synaptic efficacy at some synapses. The question is where in the trigeminovascular pain, which brain region, probably in many brain regions. And what this paper, the contribution of this paper is that it shows that the cingulate cortex is really a very important one.
The other aspect of this, which is not part of your paper, but I know is an area of research for others, has been also the whether drops in estradiol are playing a part in this central sensitization, because this hormone has a protective function for nerves. And although I don't know the exact mechanism, clinically what I see is that this central sensitization model is predominantly strikes during the perimenopausal period in women's lives, when their estrogen levels are dropping.
That's very
more and more complaints about all kinds of sort of aberrant special sensory sensations, including, the sinus pressure, but also that's when they begin to have their olfactory system and their vestibular systems and their auditory systems seem to be going rogue on them. So again, we can't draw any of these conclusions, but but this is intriguing to me, whether it might be a piece in the puzzle.
Well, I am not an expert on hormonal effects, but I know that they affect very much the glutamate handling, both at the level of receptors, at the level of release, at the level of reuptake of glutamate. Actually, I read a paper recently showing that in a chronic model of migraine, which considered to be a chronic model of migraine, which was obtained by repeatedly applying an inflammatory soup to the dura, there was a reduced uptake of glutamate in the trigeminal nucleus caudalis. So again, pointing to, there was obviously central sensitization and pointing to a role of excessive glutamate in central sensitization. So excessive glutamate has a role in both, and impaired glutamate handling has a role in initiation of cortical spreading depression, but also in central sensitization.
So I'm going to now shift to maybe underline what might be interesting to somebody who wants to get into this field of research, and that is to explain some of the techniques that you've used. So there's a term, whole cell electrophysiology. Is there an easy explanation of what that is?
Well, yes. This is a technique that allows to measure ionic currents produced by the flow of ions across cellular membrane at the single cell level, which obviously are mediated by specific ion channels or transporters. In this study, we use it to actually measure glutamate uptake into astrocytes, because this creates a current, and we measure the current, which is mediated by specific glutamate transporters. We use it also to measure the activation of specific glutamate receptors in the postsynaptic neurons following stimulation of synaptic release of glutamate. A different configuration of the same technique, which is the patch clamp technique, allows to measure the changes in membrane potentials that are a consequence of these ionic currents. So in other words, to measure the use of the electrical signals, action potential, postsynaptic potentials, with which neurons communicate. And in this study, we used this configuration to measure the so-called, which is a little something not usual, the so-called NMDA spikes, which are special, very prolonged action potentials that are generated in the dendrites of pyramidal cells when a sufficient number of these specific particular types of glutamate receptors, that are the NMDA receptors, are activated. And these NMDA spikes, they really are considered very important for neuronal physiology because their generation strongly promote firing of pyramidal neurons, firing mediated by the usual brief sodium channel mediated action potentials. So these NMDA spikes in the dendrites promotes high frequency firing, usual firing activity of the pyramidal cells. And in this paper, we show that impaired glutamate clearance results in facilitation of the generation of NMDA spikes in the dendrites of pyramidal cells, and hence in increased firing, high frequency firing of the pyramidal cells in the cingulate cortex.
So this is a explanation of hyperexcitable state is that correct? Just to simplify.
Generation of these special spikes, which then translates into high frequency firing of the pyramidal cells.
So thank you for that explanation. And how do you use a 2-photon microscopy in a study like this?
Well, we use 2-photon microscopy to actually measure the time course of extracellular glutamate upon synaptic stimulation in slices of cerebral cortex using a fluorescent glutamate sensor. This is actually a recently developed genetically encoded glutamate sensor, which means that it is a protein which changes its fluorescence spectrum, its fluorescence upon binding of glutamate. And being a protein, we can express this sensor with the modern techniques, either in neurons or in astrocytes. The binding site for glutamate, this is a membrane protein, the binding site for glutamate is in the extracellular phase of the membrane. So we can detect, by measuring the changes in fluorescence, we can detect the changes in extracellular glutamate during neuronal activity. And this is what we have done. And what we have shown is that the glutamate increase is prolonged in this genetic mouse model of migraine, reflecting and confirming the impaired glutamate clearance that we had measured with the electrophysiological technique, as I told you before, by measuring the glutamate transporter current. So both techniques give the same conclusion.
And then there's the astrocyte gene rescue.
Yes. So as I said before, the migraine causing, as a consequence of the migraine causing mutation in the gene encoding this specific sodium potassium ATPase, the protein, these ATPase, the expression of this protein is reduced to half in the astrocytes of our mouse model. And we can correct this defect by expressing the wild type protein, the normal protein, an appropriate amount of the normal protein in the astrocyte using a viral vector containing the gene for this protein. And so that we compensate the defect, because we bring up again the concentration of the ATPase to the wild type level. And in the paper, we, Mirko and Jennifer, did that by injecting the viral vector, which is targeted, the expression of the protein can be targeted specifically to astrocytes by using an appropriate promoter in this vector. And what Jennifer and Mirko did was to inject the viral vector in the cingulate cortex of the knock-in mouse, the genetic mouse model. And in this way, they rescued the defect specifically in the cingulate cortex, not in other, because obviously the defect, the impaired glutamate clearance is all over, but they rescued it specifically in the cingulate cortex. And they showed that by rescuing the ATPase expression, they rescued also the glutamate clearance, the NMDA, the facilitation of NMDA spikes, the increased firing of the cortical pyramidal cells in the cingulate cortex, which went back to normal. And strikingly enough, these also strongly attenuated the hypersensitivity phenotype to a migraine trigger that these mice, the increased pain score that these mice showed, demonstrating then that a defect in the cingulate cortex really is sufficient to alter orofacial pain sensitivity.
I understand. So they were looking at this hypothesis and testing it in a different way. And I should have you explain quickly, you've referred to the pyramidal cells. Our audience may want to know what the function of the pyramidal cells are.
Well, the pyramidal cells, pyramidal neurons, are the principal neurons, the excitatory neurons of the cortex. And in particular, all these experiments were done in layer 5 pyramidal cells, which are the output neurons of the cortex. So the so-called descending projections from the, for example, from the cingulate cortex are nothing else than the axons of these layer 5 pyramidal neurons. So showing that the hyperactivity of these neurons, increased firing of these neurons as a consequence of the migraine mutations, means to show that also you can predict that also all the downstream regions where these neurons project, the activity of these neurons will be altered, and it is known that these, these are regions mediating the modulatory pain pathways, both giving to both inhibition or facilitation. It depends. It's very the central modulatory pathways of pain, which are so important for central sensitization too.
Great, thank you. And as we're winding down here, I'm just going to, you've alluded to the fact that another lab, which had probably different expertise, was used to perform the astrocyte gene rescue experiments.
These are the people,
is this in your lab?
The first and corresponding author of the paper, obviously.
Yes.
I collaborated with them. Yes, I'm not able to do that in my lab.
Right. My question was, first of all, how many different labs were involved in this study?
Essentially 2.
2. And how often have your two labs been working together?
Well, you know, actually, Mirko Santello, who is the corresponding author of this paper, he did his master thesis in my lab, working on a project involving the another actually genetic model of migraine, the familial hemiplegic type 1 model, which is familial hemiplegic type 1 is caused by gain of function mutation in a calcium channel, resulting in increased glutamate release rather than in slowing of the glutamate reuptake. And then Mirko went to Switzerland, where he obtained his PhD, studying the interaction between neuron and astrocytes. He did also his postdoc in Switzerland, studying the mechanisms of neuropathic pain. And then he got a very prestigious grant, which allowed him to do independent research, but in a lab within an established lab of Switzerland, which is the lab of Ulrich Seilhofer, who is a co-author of this paper. So this work was done by me. Mirko and I continued to collaborate because Mirko continued to be interested in migraine. And in fact, we collaborate in other different projects. We are co-authors of other papers. And but this particular study was done so in the lab of Ulrich Seilhofer, but with this special grant that allowed to carry independent research by Mirko, and also to be the supervisor of Jennifer Romanos, who was the PhD student in the lab of Mirko. I provided the FHM2 mice and continuous discussion, obviously, design of the study and so on. So this was the type of collaboration, but all the experimental work has been done in Switzerland.
Are there further follow-up studies that you think you're going to work with this team to other questions to answer, or are you?
Yeah, there will be very interesting study to do, an obvious, but I'm not well now. There is a special situation, so I'm not sure if we will be able to do that. But it would be, for example, very interesting to study whether whether it is the anterior cingulate cortex or the middle cingulate cortex that are involved in this. And it would be very interesting, for example, to see how the rescue of the gene defect in either the anterior or the middle cingulate cortex affect the downstream regions that are involved in pain sensitization, in central sensitization, that you are so interested in.
Yes, and of course the other big leap of faith here is that you're using a pretty rare form of migraine. And the question is, what does this mean for the heterogeneous mess that we deal with, which is not familial hemiplegic migraine, but is, you know, this other spectrum of other genetic defects, mutations.
This is the big open question, how generalizable are these insights that we gain from these genetic mouse models to the common migraine. But I think I am optimistic because for example, if I put together this study together with many other studies that I have done with these genetic mouse models, really the insights that you get, all the studies indicate that an imbalance between glutamate release and glutamate reuptake, and as a consequence, excessive activation of NMDA receptors are keys in the pathogenesis of this rare form of migraine. So how relevant is this for the common form, as you say? Well, there are clinical studies pointing to an impairment of glutamate handling also in migraineurs. And you probably are well aware of recent, they are small, but they are double-blind, placebo-controlled small trials showing that memantine, which is an open channel blocker of NMDA receptors that probably has a specificity for some subtypes, seems quite effective in migraine without aura, so common migraine. And you know, the problem with drugs against NMDA receptors is usually the lot of adverse effects that they are, because NMDA receptors are very important for many functions, including learning, memory, and so on. But we have data, unpublished data, showing, for example, that in this genetic mouse model, the NMDA receptors that are crucially involved in facilitation of cortical spread in depression are a particular subtype of NMDA receptors. So perhaps, you know, if there was a drug that it was specific for this particular subtype, the adverse effect would not be so much. And this might perhaps also explain the fact, if memantine acts specifically on some subtypes, why actually it has so little adverse effects, in contrast with ketamine, which ketamine instead, as you know, has a lot, which has been very effective against depression, but has a lot of psychotomimetic adverse effects.
And how is the general climate now for migraine research in Europe? We struggle in the United States to attract people into this field. Do you have the same problem in Europe? And is there
problem?
financing?
And moreover, we have the problem that it's not, there is no money for this disease in Europe. At least in the States, I know that.
Well, there's been a lot of advocacy work to focus this with the various branches of the NIH, and in fact, there are some real advocates now within that field.
There was also a program, a specific grant money dedicated to it, right?
Yes.
There's nothing like this in Europe. It's really very depressing in this sense.
Well, I'm sorry to hear that, because, but this is, you know, what we're hoping to do is to find ways to make this career an attractive one for young scientists. So we'll have to continue thinking big about how to collaborate and finance the next steps of research. I thank you for your time and your excellent explanations, and working on these issues further with you.
Okay, thank you
so much.
it was a pleasure.